Good morning, everyone. Firstly, I would like to thank the BSN Society to give me the opportunity to take part in this Congress. And I'm delighted to present you my thesis subject that concerns the effects of GAT65 specific antibodies and more precisely B78 antibody on synaptic plasticity in hippocampal organotypic slices. So, as you know, the synaptic plasticity is the ability of synapses to modify their efficiency after neuronal activity. LTP, for long-term potentiation, is one kind of synaptic plasticity that consists in a persistent increase of synaptic transmission, and this phenomenon constitutes a molecular model of learning and memory. In this context, I study the ARC protein for activity-regulated cytoskeleton-associated protein that is specifically induced in activated dendritic spines. So the mechanisms of its expression are poorly understood, and very little is known about a possible role of GABAergic neurons in this regulation. Nevertheless, some papers show a relationship between GABAergic neurons and the ARC expression. To investigate this interaction, GAT65 was targeted because it is a characteristic enzyme of GABAergic neurons. This one is the smaller isoform of glutamate decarboxylase that transforms glutamate to GABA in synapses. It is important to know that GAT65 is the target of autoantibodies in various neurological disorders and also in type 1 diabetes. True GAT65 specific human monoclonal antibodies, B78 and B96.11, were characterized by the Professor Amp of the University of Washington. This picture shows targeted epitopes where B78 antibody binds the carboxy terminal domain, of GAT65, while B96.11 antibody binds the PLP domain. Previously, some studies were realized on these two antibodies in the cerebellum, and these have demonstrated that antibodies reduced enzymatic activity of GAT65 and produced deficits in motor and cognitive functions with always a higher effect of B78 antibody. This result is explained by an interference of B78 antibody on the association of GAT65 to GABA vesicles, so disturbing GABA exocytosis in synapses. So given that some studies have demonstrated negative impact of these antibodies in the cerebellum, we wanted to investigate their potential effect in the hippocampus that is crucial for learning and memory. My experimental model is the hippocampal organotypic culture. I use newborn rats from which brain is removed and hippocampi are isolated. Every hippocampus is cut into slices of 400 micrometers, and they are maintained in interface between medium and air using adapted culture inserts. Here is an example of one hippocampal organotypic slice where you can identify the dentate gyrus and the cornu ammonis like in acute slices. So the first result concerns the location of antibodies in slices after incubation. For that, immunoglobulins were previously conjugated to a fluorescent molecule. These pictures show that antibodies are inside cells, suggesting that immunoglobulins are internalized by living cells. I have also used a control antibody, HAA1, without GAT65 specificity, where also an internalization was observed. After having demonstrated that antibodies are internalized by living cells, cell viability was investigated. These box plots show an increase of cell death over time, where no difference was detected between different antibodies at the higher dose of 100 microgram by milliliter. To know if this increase of cell death was produced by antibodies, a comparison was realized with control slices without immunoglobulins, and no difference was detected, suggesting that this increase of cell death is natural in culture, in my model, and it is not induced by immunoglobulins. The macroglial expression was also investigated in slices incubated with antibodies during 3 days. These histograms show a significant macroglial proliferation only with B78 antibody from 0.5 microgram by milliliter. From a morphological point of view, we have noticed that microgliocytes had ramified activated phenotype, and no macrophagic cells were observed like in a control experiment. Now, in the context of synaptic plasticity, LTP was studied in slices incubated at the lower dose during 3 days. LTP was induced by SKF perfusion, followed by 3 trains of stimulations. And this result is very interesting because even at the lower dose, B78 antibody in red impaired completely LTP, contrary to the other groups for which EPSP could reach 250% of the baseline. The ARC expression was also investigated because it is a crucial protein for synaptic plasticity. Given that ARC protein is not expressed at the basal state, it must be induced. For this, I use forskolin and IBMX because it is known that these drugs increase the intracellular AMP cyclic AMP in cells. So this Western blot shows that the expression of ARC protein increased significantly in control slices after this induction. However, B78 antibody abolished this induction, contrary to the other groups for which ARC level increased about 200%. Given that ARC protein is also implicated in dendritic spine structure, I wanted to investigate if B78 antibody could also disturb dendritic spines. For that, neurons in my model were transfected with EGFP by bioelastic method, and then 3D reconstruction was realized for every transfected neurons by IMARIS. The spine density was determined in control and B78 groups where no difference was detected. The same analysis was reproduced only on apical dendrites because they are stimulated and recorded during previous electrophysiological studies, but again, no difference was observed. Then spines were classified into 3 groups in function to their morphology to quantify proportions of immature and mature spines. And you can see that the numerous spines are mature mushroom type, contrary to immature stubby and thin spines. And statistics reveal no difference between control and B78 group. However, it would be very interesting to reproduce this experiment after ARC induction to know if B78 antibody could disturb spines during synaptic activity. Now, to sum up, we saw that GAD65 specific antibodies are able to enter living cells without to affect cell viability, that only B78 antibody activated the immune system, so producing a significant macroglial proliferation, that B78 antibody inhibited LTP and also ARC induction, and finally, it didn't disturb dendritic spines at the basal state. So in perspective to this work, it would be attractive to elucidate interactions between GABAergic interneurons and microglia in the context of synaptic plasticity. To conclude, I thank you very much for your attention, and also I want to thank my colleagues. Thank you. I don't have precise response to your question because I don't investigate this topic, but I have maybe an idea. I think that given that GABA exocytosis is disturbed with B78 antibody, and that GABA is known to have anti-inflammatory effects, maybe macrogliocytes having GABA receptors could sense GABA level in synapses, and so when the level of GABA decreases, maybe macrogliocytes become activated and proliferate, but it is just an hypothesis at the moment.
And do you know if it is reproliferation you see, or just like an activation of macroglial cells?
It is a proliferation of activated macrogliocytes because their phenotype is similar like in control experiment, for example, but the number and the surface of macrogliocytes increase significantly from a lower dose of 0.5 microgram by milliliter. So the interaction between antibodies and ARC expression? it is now in the scientist literature that GABAergic neurons, that there is a relationship between GABAergic neurons and plasticity, synaptic plasticity, for example, on ARC expression, but the mechanisms is still poorly understood. I know a paper, for example, that demonstrates that reelin, that is a molecule secreted by GABAergic neurons, has a positive effect on the ARC expression, but a mechanism mediated by GAD65 is still unknown at the moment.